Okay. So I'm going to begin. We are going to begin. Bueno, buenos días a todos por los que están aquí. Les doy las gracias por no estar viendo el partido. Y como saben, como lo dije anteriormente, es el último seminario del programa de Frontiers in Genomics de este semestre y por lo tanto de este año. Y como saben, es organizado por la licenciatura en Ciencias Genómicas, el Centro de Ciencias Genómicas y el Instituto de Biotecnología de la UNAM. Como siempre, las preguntas las pueden formular al final, pueden poner la manita en el chat y yo les doy la palabra, o pueden inscribir la pregunta en el chat como tal. Bueno, vamos a comenzar. Para mí es un gran gusto presentarles a la doctora Julia Zeitlinger del Stowers Institute for Medical Research. La doctora Zeitlinger hizo su licenciatura de 1991 a 1995 en Biología Humana y se recibió con honores en el King's College en Londres. De 1995 al 2000, Julia hizo su doctorado en el grupo de Dick Borkman en el MBL, obteniendo el grado por la Universidad de Londres, trabajando con el oncogene FOS en Drosophila. De 2000 al 2007, hizo estudios postdoctorales en el grupo de Richard Young, que es una de las luminarias en el campo de transcripción, en el Whitehead Institute for Biomedical Research en el MIT, donde trabajó sobre redes transcripcionales en levadura y Drosophila. Del 2007 al 2013 fue Assistant Investigator en el Stowers Institute for Medical Research. Desde 2008 al 2015 fue Assistant Professor en el Departamento de Patología y en el Laboratorio de Medicina en el Medical Center de la Universidad de Kansas. Desde 2013 al 2018 investigadora asociada también en el Stowers Institute. Y del 2015 a la fecha también es profesora asociada del Departamento de Patología y del Laboratorio de Medicina del Medical Center de la Universidad de Kansas, y a partir del 2012 es investigadora titular del Stowers Institute. Durante su carrera, Julia ha recibido distinciones importantes, y solamente por el tiempo voy a mencionar algunas. En el 2015 recibió el News Award Stowers Institute for Medical Research. En el 2012 fue Hudson Prize por la Fundación Hudson, por la Fundación Hudson. Del 2008 al 2013 obtuvo el Director's New Innovation Award del NIH. Desde 2008 al 2012 fue Pew Scholar en Biomedical Science, lo cual tenemos algo en común en eso. Desde 2002 al 2003 fue Fellow del programa de Human Frontiers. Desde 1995 a 1999 fue estudiante distinguida en el MBL, y en 1992 al 95 recibió un German Academic Scholarship Foundation. Ha recibido donativos múltiples de muchas instituciones que incluyen el NIL, National Human Genome Research Institute, y de la Pew Foundation. Es revisora de los journals más importantes en nuestro campo, como son Nature, Science, Nature Genetics, Molecular Cell, etc. Y el día de hoy nos va a hablar sobre High Resolution Genomics to Understand Transcription in Vivo. Thank you very much, Julia, to be with us. So it's an honor for us to have you here in this program, and now we are ready to hear your talk.
Okay. Can you see this?
Yes, we can see it.
All right. Well, thank you so much for having me. And especially, I know there's the soccer game right now, so I'm very honored that you could attend this talk. And I'm just going to get started. So my lab is interested in understanding development. And ultimately, we know it's a very reproducible process that's encoded in DNA sequence. So it's a wonderful problem that is known for a long time, and yet we cannot really decode the information of gene regulation by sequence alone. So gene regulation has been studied for a long time, and we know a lot about it. We know the transcription factors are reading out these cis-regulatory sequences by binding to short motifs, but we don't know what exactly about the combination of these motifs makes the enhancer active and therefore produces these beautiful patterns, let's say, for example, in the embryo. And now, of course, we live in an era where this should be a problem that is addressable. So right now, with an explosion in genomics technology, we have now data on how transcription factors bind in vitro, for example, Celix data. We have data on how transcription factor bind in vivo or on chromatin accessibility. We have data that are a little bit more related to enhanced activities such as histomodification or enhancer transcription, or even directly reporter assays such as StarSeq. We can also look in the 3D organization of the nucleus, which hopefully will help us to relate which target genes are activated by enhancers, such as high-C data. And of course, we can map transcription, even nascent transcription, at really high resolution, including single-cell resolution. And we can even look at proteins. We can also look at perturbation, such as CRISPR screens, but also we have lots of information from GWAS. So all of this is really wonderful, but there is this big question. How do we make sense of all these data? How do we put that really together? And I think there is a little bit of a sense in the field. There's lots of technology published, but what are we really learning from that? And so one of the sentiment is I quoted here by Sidney Brenner, which is, technology gives us the tools to analyze organisms at all scales, but we're drowning in a sea of data and thirsting for some theoretical framework with which to understand it. So why is that? And so I would argue here that we do have a framework, and we just have to get back to that and have to develop the right tools to really look at that. But I think the time is right. And the idea is simply to look back at DNA sequence. Oops. So we know already since the 1950s that almost any biological process has a basis in DNA sequence. And so this was already discovered in the 1940s by Oswald Avery, where he found that the transforming agent of a bacterial disease was really the DNA. And that was really surprising. And in fact, he wasn't believed at the time until almost a decade later, the phage group also showed that really the material of inheritance was DNA. And this was also the decade where the structure of the DNA was discovered by Watson and Crick. And of course, that provides a fabulous carrier for information, genetic information, and that can be read and copied. And at the same time, Drosophila has been, of course, a tremendous model organism for a long time. It became also clear that genetic information drives development, and that information can be changed, for example, by X-ray mutation. So we can alter that information and we can alter development accordingly. So all of this gives a very clear picture that that information should be ultimately linked to DNA. And we are exactly in that same situation today, where it's sort of almost screaming at us. All these GWAS studies really saying there's lots of information that we cannot quite decode, but we know it's contained in the information, it's in the DNA sequence. So a lot of disease susceptibilities, if we would understand them, it would really help us to take better care of ourselves, for example. And so I think there is really a big incentives to better understand that, but it's also a hard problem, of course. We also have the ability to read this information. Now with sort of the $100 genome approaching, I think it's pretty clear that in our lifetime, we are able to access DNA information quite easily. But yet, can we extract the information that's really in there? We also have the ability to change DNA by editing. So that's, of course, almost a little scary, but it does provide also incentive for therapeutic treatments. So all of this is really saying DNA is the key, but how do we really read this information? And I would argue here that it sort of has failed on the more traditional side to really dissect it one at a time. But there are now tools available to do this in a different way. And this is using neural networks to understand DNA sequence information. So you may already know from social media when you, for example, look at your iPhone, the iPhone is really good at recognizing faces. And this is just part of sort of the revolution of using neural networks to understand patterns and give you that, make that information available to you. So why is it so powerful? Because it can learn patterns in a really unbiased way you need a large amount of data, but it can recognize pattern really well. And it can be trained to predict those patterns that it seems almost impossible to do with the human mind. So as an example, back here, you would find pictures of me, but it's completely different backgrounds with glasses. And yet the neural network is really good at identifying faces. So why is that? So when a neural network is trained to predict faces, it learns this in a really unbiased way. So it's trained on recognizing faces from a large amount of images. And it does so from the bottom up. It will look initially for very small pixels that form edges, let's say. Then this is put together as a larger object, such as eyes, nose, etc. And then eventually this is also put together as a face. So it's sort of a hierarchical model. And any structures that are helpful for the predictions will be learned. But initially it's completely unbiased. We're starting with random seeds. And so then, of course, this can be applied to specific faces. So this allows us to learn from very sparse data in a completely unbiased way. And what's so cool about that is that can also be applied to DNA sequence. And this has really changed the way we do genomics data analysis. So I'm traditionally a developmental biologist, but when I came across using neural networks, to me, it was immediately clear that this is a game changer. So because traditionally, and we've been doing this for the last 20 years, when we analyze the genomic data sets, we're taking it apart. So let's say we're taking a transcription factor bound to DNA sequence, it's like a ChIP-seq data set. We're taking all the bound sequences and looking for patterns that are enriched. So we're hypothesizing that motifs are enriched, maybe even motif combinations. And we're looking for overrepresentations. And so that would be an indication that the pattern is present. But the problem with that is that once we have these sort of abstract representations of patterns, we can't really put it together. So we can't really say, well, whenever we see that pattern in the DNA, is that really a bound region? It's not a very good method of explaining all the bound regions. So often you find regions that have low affinity sites that don't even have a motif at all. So they're not very good. They're not predictive. Now with neural networks, it's the exact opposite. So that's why I'm calling it the inverted learning paradigm. We're training initially for high performance. We're not putting any hypothesis in. We're not even putting in that there have to be motifs in there. Well, all we're giving it is genomic regions. In this case, is 1 KB, but it could be extended, of course. And we're saying from only DNA sequence, predict us the genomic data sets, let's say bound transcription factor. And so now the neural network has to learn from the bottom up. So learning motifs, like this is the small patterns, and then putting those motifs together in a combinatorial way to predict these bound regions. So we're not asking it to learn specific rules. We're simply scoring when it performs well. And they do. That's what they're trained to do. They're highly capable of very high predictive accuracy. And so now we're turning this problem around. We're having high predictive accuracy, but we don't know how it did that. And of course, that's key, because just predicting data that you already have is not helpful. Instead, the challenge is to look inside that black box and to understand the rules by which the network was able to predict. And of course, we do know that it has learned general rules, because it can't just memorize the data, because we're testing it on data that the model has never seen before, on held data. So the rules are general, but we need to extract them. The good news is that DNA is quite simple, much simpler than images. And so therefore, it's actually easier to extract the rules than from images. It doesn't mean we can necessarily extract everything all at once, but it's actually very powerful. And I'm going to show you just a little bit of how that works. In the worst case scenario, we can always use the trained model as an oracle. We can say we don't completely understand how the predictions are made, but we know they're really good. And so we can take, for example, different regions where we perturb the sequences, we're mutating, let's say, binding motifs and asking, how does this affect the predictions? We can also do this, for example, for cross species. If we're taking sequences from different species and ask, how does that perform? Or genetic variation, disease variants. And the model is very accurate in predicting whether this disturbs function, even if we don't understand the rules. So those are the sort of two powers of neural networks: interpreting the rules that the network learned in an unbiased way and using the accuracy to predict the effect of sequences that are of interest. So what is the requirement? Of course, not all models are the same, and that can be confusing when you look in the literature. We want a model that is well trained, of course, that's highly accurate and therefore much more likely to really extract the biology that we're looking for. And we need good interpretation tools. And so there are a couple already available, but there's certainly more to be done. Now, we got into this because we like genomics technology, and our lab is really specialized in going the highest resolution. For me, this was important because I always felt that we can't quite know the cis-vectorial code if we don't know the exact motifs that are bound. So to start, we have to map the motifs really precisely. And so my lab developed this technique called ChipNexus, where we're pulling down a protein or a transcription factor of interest, and we're pulling out the DNA, but we're digesting the DNA with an exonuclease. And so therefore, we're getting much higher resolution. In fact, we're getting these footprints, depending on which strand, and either from the forward or from the reverse strand, we're getting these footprints, or here shown in red and blue. And so this is one transcription factor or two transcription factors here from mouse embryonic stem cells. And you can see there's a very consistent footprint. So that really should allow us to recognize where exactly transcription factors bind. And the resolution is much higher than ChIP-seq data. It's also of higher specificity. For example, OCT4 binding at the SOX2 motif is much lower in background in the ChIP-Nexus data than in ChIP-seq data. But once we had the data, we were also facing the problem that we didn't know how to really extract the sequence information out of there. And so this really led us to a wonderful collaboration with Anshul Kondaj's lab at Stanford, who had already started using convolutional neural networks to help understand regulatory information. And it turned out that the combination was really powerful because it forced them, or sort of we asked them to learn these data at base resolution. And this was not before, images are usually classified into yes, it's there or it's not there. Whereas now we're asking the network to predict data in a quantitative way. So just to summarize this part, so we have highly quantitative information, the highest resolution on transcription factor binding, and we're now adding a neural network that predicts at base resolution. So here, and I'm just going to show you here, this is the observed data for one transcription factor OCT4. Here we have a footprint in the middle, and the network is able to predict pretty much that exact profile. In fact, it denoises even a little bit, which is the really great power of these neural networks. So we're not just doing classification, we're predicting the read counts, so the total amount of reads, but also the exact sort of the distribution of these reads across the entire region. And that forces the network to learn really precise sequence rules. And so it turns out, even if we apply this to other data sets that don't have quite the resolution of ChIP-Nexus, it's still very, very powerful. And so that's why Anshul and my lab have kept this BPNet approach of predicting at base resolution, even for other data sets. And so rather than, we've kept that, but we've added, for example, correcting experimental biases, adding new interpretation tools, et cetera, et cetera. So that's work ongoing, and I'm going to show you a little bit of that. So what do we do with that? So of course, the big question has always been in the field, does it matter how the motifs are arranged for getting enhanced activity? So in the field, it's been sort of a pendulum between extreme views. So initially, people thought of the combinatorial code as proteins coming together, binding to DNA. And so I think the initial impression was that this is sort of like Lego bricks, and these proteins are interacting with each other. And there was some initial data also showing that if you change the spacing of binding sites, that actually disturbs the enhanced activity. And so this has given the impression that there is very strong syntax. So syntax is the arrangement of the motifs, plays a major role in the enhanced activity. But subsequent experiments have sort of suggested the opposite and said that, well, it doesn't really matter that much how these motifs are arranged. So each motif is sort of an information display, like on a highway, but the combination, the order of it doesn't really matter. So in this case, you would say there's no syntax. Now, of course, the truth, as always, is usually in the middle, and we're not the first one to proposing that, but we did see kind of a really nice way of summarizing that, which is kind of a motif function. So first of all, we did see strict motif syntax, and that wasn't surprising. So sometimes we do have proteins that are coming together on DNA, and they stabilize each other's binding, and that is indeed strict syntax. So there's a very precise spacing, in this case between SOX2 and OCT4. But we also saw interaction between transcription factors that we call soft syntax, where it's sort of a distance function that determines how much increased binding a transcription factor gets through another transcription factor. So we call this cooperativity that's distance dependent. So it is not all or none, but the amount of boost depends on the distance. In this case, there's also periodicity, as I'll tell you in a minute. And we also see something interesting in that often there is directionality. So one transcription factor benefits a lot more from the other than the other way around. And so this is really interesting. And so we think it has a lot to do with chromatin accessibility, as I'll show you in a minute. So today I'm going to talk more about this, but just to give you a brief kind of insight in how this actually works. Not a lot of technical detail. I know it can be confusing, but just kind of a little bit of information of how we're extracting the information. So initially, we're training the model. So this is just, this is sort of the bottom. The input is the DNA sequence. This is how it's represented. And so then we have these layers. And so this is deep learning because we have multiple layers, which gives the network the ability to not only learn motifs, which are, networks are great, but it also learns to combine the motifs into some higher order combinations. And then, of course, it learns to make footprints that are specific for the experiments. In fact, very easy to learn for the network. So once we have that, and we have high predictive accuracy, as I've showed you already an example, but it's pretty amazing, I think, that it can do so well. Now we're going in and interpreting the network. So we're opening the black box. And so the easiest way to do that is to simply trace back the predicted output to the input, which was the DNA sequence. So this is kind of, sort of imagining like that. It's an algorithm called SHAP. And DeepLift was the implementation from Anshul's lab, but more generally it's called SHAP, where it kind of makes a decision of how to distribute the importance of these prediction to the input bases. And so it gives this what we call a contribution score for the bases. And what's really nice is that we can immediately see that we get high contribution for motifs. The motifs are really kind of magically appearing already if we're simply doing this very systematic backpropagation approach. So this is what one region looks like. So we're clearly seeing that these motifs are showing us they are important. So we're just looking at each base, but we're seeing motifs already. And we're seeing that these motifs are different for different transcription factors. So NANOG, for example, also likes this motif. It's an OXOX motif, but it also depends on this motif where SOX2 is not. So this shows us immediately that there's cooperativity, that NANOG is dependent on not only on its own motif, but also on other motifs. And most importantly, what I'm showing you here is just one individual region. So rather than learning an abstract representation, we are learning really the contribution of each individual region. So it's based on that, and each individual region performs well. So it's the opposite. We're going from each individual case, and this is where the precision is. But now we have to, of course, also learn what are the abstract representations. And so I'm going to skip through that, but we can learn motifs, of course, and then go back and label these genomic instances with the correct names. And so once we have that, we can also do more systematic perturbations where we perturb motifs and then ask what happens. So again, we're using the trained model as an input and then ask what happens if we perturb a motif, and let's say what happens to non-hoc binding. So this gives you sort of just an impression of what we're doing. Like once we've learned the network is trained, what kind of information we're getting out of that. So these sort of contribution scores is a lot of what we're using, because that's sort of the raw output of what the model learned and what we're interpreting as being important. So just to mention what's really cool about neural networks is that they discover patterns that we didn't necessarily hypothesize. And so, for example, non-hoc is a really well-studied pluripotency transcription factors. Lots of genomics studies have been performed on that. But a lot of studies have initially or had identified the OXOX motif as the most overrepresented motif. But we see very clearly what the non-hoc motif is. And that's very different from that OXOX motif. And we also see, and this is the CWM is not a PWM, not based on frequency, but based on the importance for the network. And we can see that there is, in addition to the central motif, there are these flanks that seem to be really important for non-hoc binding. And so that is of 10 base pair periodicity, which is really cool because that has been proposed to be part of the combinatorial code for a long time. And so we have a really nice example where this is indeed the case. So non-hoc can multimerize and can bind presumably to these sort of longer stretches of DNA. So here we see regions where we see these sort of important scores. So that's kind of what we would deduced as important for the binding in these regions. And this is where we see this periodicity. We also validated this. This is just one example where, again, we're showing the predicted profiles by the network. These are the actual observed chip nexus data that we did in wild type. And also when we CRISPR mutated the non-hoc motif, and we can see that the binding footprint on this motif indeed disappeared, showing that it is the non-hoc motif. And we also see its effect sort of on the neighboring sequences. So there is indeed kind of a little bit of a long range effect of an interaction that we did extract from the model. So this was nice to see. But now today I'm going to go into some newer work. So this was published work, but it gave you sort of a little bit of an example of how we're extracting the information. Now we're taking this to a new question, and that is chromatin accessibility. Chromatin accessibility is a really popular readout because it's easy to do. So ATAC-seq data, for example. But biologically, it's actually kind of an interesting problem because we know we need chromatin accessibility for enhancers to become active. And that's not trivial because, yeah, so we don't understand fully the relationship between transcription factor binding and accessibility. So if you go to the literature or to talks, let's say on conferences, there's sort of different takes on that. So some people see chromatin as almost separate to transcription. So I guess something that's, you know, to do with memory, with developmental history, perhaps. And so it may not be exactly related to what's happening here. So this would be saying it's dependent on developmental history. Or then, of course, in a more transcription field, they would say, well, we know that the dedicated transcription factors that open chromatin, so-called pioneer transcription factors. And so therefore, if you look at accessible chromatin, you really should be looking at pioneer transcription factors. However, if you've ever analyzed ATAC-seq data, you'll see that there are lots of motifs that are enriched. And so often the takeaway is like, well, we see really almost any transcription factors. So what is it? And so we were really interested in using the BPNet approach to dissect what is it, what we're really seeing with chromatin accessibility data. And especially having really strong transcription factor binding data. So when we can directly compare that, we should be able to extract what we're in fact seeing with chromatin accessibility. And so the power of neural networks in this case is really great, because when you use a traditional approach and you simply look at accessible chromatin region and you look for motifs, yeah, of course, you see a lot of enrichment of motifs, but it doesn't really tell you the causal relationship. Because you don't know whether the motifs that are found there are causative for opening these regions, or whether those are the factors that help activate the region once the region is open. But because BPNet learns these regions in a much more quantitative way, and it learns motif combination, we have a much better shot at deriving causal relationships. So for example, you can easily sort of by intuition see that if we're looking at open regions and then we're looking at all the motifs that are found there, you would see a much better correlation between the pink motif and accessibility than the other motifs. And so the neural network would simply try to predict these open regions and therefore infer the motifs that are most predictive. And so this is really powerful. And so we wanted to apply this approach to predict chromatin accessibility. And we wanted to do this in the early Drosophila embryo because, well, first of all, we have studied that, but it also is a really powerful system for understanding enhancers. And there's lots and lots of information, genetic studies out there that gives us confidence of what we're seeing is indeed real. And also, we have antibodies, we can do chip nexus on those transcription factors. And it was really kind of a challenge to see how well we can perform if we're mapping transcription factors and really comparing this to all these old mapped enhancers and how well we do. And, I'm going to cut a long story. So we did actually really well. So this was nice to see. So we did chip nexus on 6 transcription factors. So Zelda and Gaga factors are 2 pioneer transcription factors. So we think that they're opening chromatin. And then we have 2 anterior posterior patterning transcription factors, bicoid and caudal, and then dorsal and twist who are involved in dorsal ventral patterning. And when we trained the network, the BPNet network, we're getting exactly the motifs that we expected. We can also see that we see very nice footprints on the motif, of course, as expected. And now we can ask, how do these factors influence each other? So do they cooperate in binding? So that's sort of the big question that previously people couldn't really address very easily, at least not sort of computationally. And what we see here, again, we're extracting the contribution scores, like how important are the motifs to each other for binding? And what we're seeing is that the Zelda motifs, so these are the motifs, and this is the transcription factor binding patterns. The Zelda motif is important for all the transcription factors. So this is consistent with experimental data that show that Zelda indeed, so in a Zelda mutant, binding of dorsal, for example, is really, really impaired. And that has also been shown for bicoid and twist. So this is very consistent with that. Surprisingly, GAGA factor is also supposed to be a pioneer transcription factor, but it doesn't seem to help the binding of the other transcription factors. We also see cooperativity between dorsal and twist, which was nice. That's also consistent with, because we know that they're working together. So in summary, we did see, in fact, the known motifs. We can look at known enhancers. I'm going to show you one in a minute, and really recapitulate what's known. Okay, so we have this foundation. We have mapped motifs. Again, the strength of the neural network is that these are not abstract motifs, but we can map them very precisely to genomic regions. So these are not just sequence matches, but truly predicted contribution to what we trained the network to do. Okay, so now with having that, we also did an ATAC-seq time course where we collected, in four 30-minute windows, embryos, while the genome is being established. So the zygotic genome is being activated. So this is nice. We're starting transcription from scratch. And so it allows us to really kind of see how this happens. And so now we had the challenge, though, if we wanted to learn ATAC-seq data at base resolution, we wanted to do a bias correction. So normally, ATAC-seq data is often kind of smoothed out. This is a coverage where you present the entire fragment, and that kind of smoothes out the bias that's inherently present in the data, because TN5 has an insertion bias. But if we want to take advantage of the base resolution and really just map the cut sites of the TN5 itself, this is what it looks like. And this is not chromatin accessibility. This is really just the bias of the TN5. So it's an experimental bias. But with innovation from Anshul Kandaj's lab, they came up with a way of really, really nicely getting rid of this bias. So we're essentially training another BPNet model simply on the bias, on closed regions. And then we're freezing that and training a second model then on the biology. And that worked really well. So then we're getting rid of the bias. So we're predicting it with a bias, so it can learn that really well. And now we're taking the bias away. So now this is what really the chromatin accessibility should look like. Well, how do we know this is good? Well, if we're looking at motifs, so first of all, we're predicting what is important for the contribution. We're seeing, clearly, again, these are these so-called contribution scores that are directly for each region. And we can see this, in fact, as Zelda motifs, but we're also seeing some of these other motifs. And when we look at the motifs, we can see now that they're sort of flanked by open chromatin, which you would expect. So for example, here, we see that. And then we also have done other interpretation tools to visualize, and the bias is really kind of amazingly gone. So based on that, we're very confident that the model is able to learn the ATAC-seq data here without the bias. Okay, so now we're going back to what is the contribution of each motif. And we did use the motifs that we identified from the ChipNexus data, just to be confident. Although we did identify the motifs also in the accessibility model, but not quite as sharp. So clearly the model has had a little bit harder time. It learns much more complex rules, and the motifs weren't quite as good as with the ChipNexus. So we have that caveat. And this is also why we don't simply rely on the network for all our conclusions. And we really go on and do experiments with that, because, yeah. But still, I mean, the network actually did amazingly well. So we do see that the contribution now to accessibility is driven by Zelda through all the time points and also Gada factor. So this time, this is exactly consistent with the literature. These are the two pioneers that are driving chromatin accessibility. But we're also seeing a contribution from these other transcription factors. And this is sort of the question, like what are they doing? What is the role of dorsal and twist in the accessibility? And first of all, we wanted to compare more directly between accessibility and the contribution to accessibility and the contribution to binding. So now we're comparing. So remember, we have all individual regions. We have the contribution very directly from the neural network. We have completely different neural networks, one trained on accessibility, one on binding. And so now we're systematically comparing the contribution to binding versus the contribution to accessibility. And what we're seeing that for the pioneer transcription factors, there's a linear relationship where even though it's completely independently learned on different data, but they actually correlate quite nicely. And they're not only correlate, but they also are correlating with motif affinity, which is sort of shown in here. So in yellow or light green, these are high affinity. So we're seeing a stronger effect on binding and on accessibility. And that's also true for GAGA. But the other factors don't do so well. So again, consistent, they are, the motif affinity contributes to binding, but not so much to accessibility. So there seemed to be a much more complex relationship. But I was nevertheless really surprised how well the relationship held for Zelda and GAGA factor, because that suggested that the pioneering can also occur at low affinity motifs. And this to me is surprising because we're thinking of pioneer factors as reading out the DNA outside a nucleosome. And we know from structural studies that this is really difficult. Binding is really constrained. It's often only on flanks or on diodes. And so yet what we're seeing in vivo here is that accessibility almost doesn't seem to care. Whenever you can bind, you can also drive accessibility. So that was surprising based on what I knew. And so we wanted to validate whether this is indeed the case. So can Zelda drive accessibility dependent on its motif affinity? And so for that, we wanted to make sure that we're in fact measuring motif affinity correctly. So what we did is we performed experiments on protein binding arrays using Zelda. And so this is the actual experimental data. This is sort of more a p-value on that. And what we're seeing is a really nice correlation between what BPNet predicted as being most important for binding and then the actual in vitro binding. And this is something that's not the first. Neural networks seem really good at extracting motif affinities. So the motif affinities are actually learned. So this is what it learns at the very beginning in the motif scanning. And then it learns to combine that. Of course, in vivo, we're seeing a lot more going on than affinities, as we can see in vitro, but it does also have to learn the motif affinities. So that was really nice to see. And so then we wanted to validate, okay, is that really true that this motif drives a lot more accessibility than this motif? And I'm just going to show you one example. So Kaelan in the lab did the experiment by depleting Zelda from embryos. So we're doing the experiment. And we're doing ATAC-seq in the exact same way across these 4 time points. And we're looking at regions that only have a Zelda motif. And these are just examples. So a high affinity versus a low affinity. And we can see, indeed, first of all, it does increase over time. And we can see that even low affinity motifs do have that contribution to accessibility. And this is just a control for GAGA. We can also look, of course, at many regions. So this is the top 250. Again, we see the same effect. Low affinity sites do drive accessibility, but at a lower extent. So this is really cool. Now, for the remaining time, of course, there was still this puzzle left. What happens at the other motifs? What is happening at dorsal and twist, for example, to accessibility? And so one thing we wanted to do is to use these Zelda minus embryo data. And we thought, well, we know that these other patterning transcription factors depend on Zelda for binding. So if we're looking at motif combinations with Zelda, we know that binding, let's say, of bicoid, et cetera, is also affected in the Zelda minus embryo. So again, we're comparing Zelda minus embryo and wild type. So we're seeing already a loss of it when we have motifs only with Zelda. When we look at other motif combinations, we see an even bigger loss of accessibility. Again, showing experimentally that these other transcription factors also have a contribution to accessibility. So they do have a role, but it seems to be context dependent. It seems to be dependent on the exact motif combination. And this is also what the model told us. So this is one example of an in silico experiment. This is again the SOG enhancer that I showed previously, where we had all these beautiful binding sites that mapped quite nicely to known and known motifs. Now we can sort of mutate Zelda and observe in red the loss in accessibility. So this is what we're predicting. So it's really cool. We're predicting lots of loss, of course, because Zelda is important for the accessibility. But we're also seeing that loss of dorsal and here a little bit of chordal also has an effect on the enhancer. So the model also says that, and it doesn't say that for all enhancers. So it is a much more context-dependent contribution to accessibility. And so now we thought this was really cool because we know the system well. And so we're saying, okay, let's figure out what this is. So dorsal has this property that it can both activate and repress genes. We've worked on this before. Here's one example. So dorsal is present on the ventral side of the embryo, and it activates mesodermal and ectodermal enhancers, not alone usually, but it has an activating effect. But it also represses the genes that are on the dorsal side. And it does so, but with the help of a repressor called Capicua, which recruits a co-repressor Groucho. And so the trick is that when dorsal is next to low affinity Capicua sites, it's turned into a repressor because it recruits Capicua and now functions as a repressor. Now we performed experiments where we got rid of dorsal. It's called a mutant called GD7. And now we're observing what happens to the accessibility. And what's very striking is that we see the genes that are normally activated, which is in the ectodermal, mesodermal genes, they now go down in accessibility. But the genes that are repressed by dorsal, which now sort of become de-repressed perhaps, now go up or remain unchanged. So this really tells us that it's the effect on activity that matters, not so much the binding per se of dorsal. And we wanted to test that more specifically by looking at another mutant called Capicua. So Capicua, now we're not doing anything to dorsal. All we're doing is eliminating the ability of dorsal to repress, because we're mutating Capicua in a way that it can no longer interact with Groucho. And now it can no longer repress. It can still bind DNA, presumably, but it cannot repress anymore. And now what we're seeing is that dorsal should still be able to activate, but it should no longer be able to repress. So we should see the effect here, but not here. And this is exactly what we saw. So the activation is still the same. It's the same as wild type, comparing Capicua, but now those genes again become de-repressed. So this really shows that the accessibility is coupled to enhance activation, which is a much more context-dependent effect than we saw with Zelda. So to summarize that part, we saw sort of the canonical sort of pioneer factor like Zelda has a very consistent effect that is dependent on the motif affinity. So it functions upstream, it's the first factor that's there and consistently opens the enhancer. It doesn't necessarily activate. It's in fact not in itself an activator based on literature. But then it needs the function of the patterning transcription factors. And you may see here that I'm sort of drawing this as a hub, which may be speculative, but there's really nice evidence on Drosophila to suggest that it contributes. And so the reason I'm pulling this up, because this really does explain a context-dependent mechanism for activation. Once we have a lot of transcription factors, so the DNA is exposed, is available, we have enough transcription factor that gets concentrated now here due to the DNA motifs. And so that could be forming a hub that's important for enhancer activation. Of course, there are other models, but what I'm saying is that this is a separate mechanism from this kind of initial pioneering that also increases the chromatin accessibility. So the answer to the question is, okay, when we're training now in an ATAC-seq neural network, we're seeing we can train it really well. We don't have to look at developmental history. So we in fact training each time point individually, and we don't need previous time points to make any predictions. But we do see a really strong readout from so-called pioneer transcription factors. They are indeed the strongest contributors, but we also see an effect of what we think of as activators that come more downstream. And this is a more context-dependent effect. So if you look at ATAC-seq data, you will see a mix of transcription factors, and you might not necessarily know immediately whether it's so-called classical pioneer or a further downstream factor. And we're looking into that. But it's good to know that there really is a distinction between these two. So with that, I don't have much time left, but I hope it convinced you that we can use now neural networks to better understand this Satori code. And we're really using this to predict our experiments. So we're not just satisfied with these are our prediction and that's the biology, but we can now go in and test those predictions And it's really been fun to sort of combine these sort of the kind of our newly gained computational tool with the experimental tools that we have in the lab. And so, of course, the goal is to derive really general rules of how enhancer function works so that we can apply it to more broader prediction frameworks that can be applied, for example, to different cell types. So with that, I hope that I convinced you that we are on a really game-changing way of doing genomics, where we're using neural networks to help us predict information. And that's a sort of equivalent to navigation in human mankind, where, you know, back in the days, biology was done really on a lot of educated guesses, you know, like very local signs, you know, you're going to try something out. Once we had genomics data, we were able to use this information to guide our experiments already, and we're doing that. But with neural networks, it can take us to the next level, where we can predict experimental outcomes, like what if you mutate only 2 bases or even 1 base. And we can pick the experiments that we think give us really strong effects and that give us really insights into mechanisms. And so that's really fun. So with that, I'm going to thank all the lab that specifically Caleb Brennan, who really pioneered the Drosophila embryo project. And with that, I'll take questions. So.
Okay, thank you very much, Julia. Very nice work. Now the seminar is open for questions. Have to see if there are hands. Well, there is one question in the chat. Can you see?
Yes. Do you think we will be able to decipher the whole Cis-Toy code using this neural network approach? Yes, of course, I think. Well, we is the question, right? And so the question is more for me, how to do that. So clearly neural networks are ideally suited to do that, right? But the challenge with the Cis-Toy code is that it's super cell type specific, right? So we have a combination of transcription factor that's usually unique to that cell type. I mean, the same transcription factor might be present in other cell types, but in a different combination. And so the interaction could be different. And so it's not clear how to learn these rules in a more general way, because the neural networks are ideally suited to learn exactly what you wanted to learn, but it's not necessarily good at learning very abstract representations. And so in order to solve that problem, there's sort of different approaches. So one would be to say, well, we have to make better models. And so I'm talking to computational peoples, of course, to how do we make models that learn more general rules? That's one way. Another way would be to say, well, we have to acquire all this experimental data. Yes, if we have experimental data for every cell type, then we can make those predictions. And there is, of course, a lot of data around. So ENCODE data, my collaborator, Anshul Kandaja, is in fact the one who's analyzing the ENCODE data and has applied BPNet already to those. So it works really well. It's already clear that we can use this to make predictions for GWAS, and it works really well. But it's still not everything. And there's still also the problem that we're not really learning general rules. Because we're not putting it in an artificial context where we're challenging really the rules. So there's a little bit of sort of an complex, so it's not complex enough actually what we're learning, unfortunately. But I mean, it's a good start. It's a good start. And we could maybe, if experimental technologies are getting better, you know, single cell resolution and the kind of resolution that we have, it could work, right? And that's something that we've been pursuing quite a bit, but it's also a hard problem, I would say. So again, both are very hard, the experimental model improvement and the experimental data expansion. But again, it's done by a lot of people. So both of it, I think, will work in the long run. And then the third approach that we're taking is to really understand the biophysics behind that, with the idea if we understand the rules, it should help build better models, and maybe even models that where we extract all the rules and we're building them from scratch, rather than having this to learn, we can actually learn general rules. Now, is that possible? We'll see. But that's kind of what I've been working on in particular, just because that's my expertise. You know, I've been looking at transcription for the last 20 years. And we have the experimental tools. And so that's why we're looking at that. And so hopefully the three approaches together will help to really get at the oscillatory code. So I think it's a matter of time, but it's clear that the capabilities of the neural networks are there. And that's really exciting for me. It's a game changer in that way. Of course, if you're a biologist and you think, I just want to learn about my system, I don't care, then this might still be early, although you can use it for sure. So if you're willing to apply BPNet, you can download it and apply it to your data for sure. So if you're only interested in one cell type, you're certainly getting much better motifs than you're getting with traditional techniques. So it's possible, but it's not quite the magic maybe that it can be. So that's kind of my take on it.
Thanks.
Thank you. There's a question from Enrique Renault. Go ahead, Enrique.
Mm-hmm.
Thank you. It was a very nice talk. I have two questions. They are kind of simple, I think. The first one is pure ignorance. How do you train the neural network? Do you take, like, you make your Nexus chipset and then you have like a label and the sequence, and then you feed that to all your neural networks, and then it produces the motifs, or how do you train them? Or you take, like, for each training, you have to train a different network for each transcription factor or something like that?
Oh yeah, so it's a multitask model. You could also train it independently, but in this case, we are training it together in the same network with the idea that it could learn from each other. But ideally, if it's perfect, and we've tried that, you can also train it apart. It learns the same patterns. We're not doing labeling. So we are pulling out regions of interest, I should say. So we're not picking regions of heterochromatin where binding is impaired, because I think the rules are much more complex there if you in regions where you have long-term chromatin repression. So we are specifically looking at open regions, but those open regions are not labeled. So we're asking the network to learn the exact data. So for every base, what the signal is in the data. And so it learns this all at once. I mean, all at once, it's you give it a training set. Of course, you only give it a part of your regions. And it has a tuning set by which it optimizes the parameters, and then it's tested on the test data set that was held out. So that's how we know it performs well. So if it can still perform well on the held out data, then we know it has learned these general rules. So I don't know, did I answer your question? There's so many aspects to it, I know. So
yeah, well, I don't have any experience on that, so I was just curious. And I was imagining that probably labeling would be very nice because it will tell you exactly not only the motif, but who is there. But of course, we also would detect the motif as it is, isn't it? So
the motif is not, we're not putting any hypothesis in there. We're not giving it motifs. We're not even asking it to learn motif. The only thing is that the network architecture is ideal for learning motifs. So the first layer, that first motif scanning layer is made to identify motifs. Although honestly, it doesn't, we're not the size is 25 base pairs initially, because we thought that would sort of cover motifs very nicely, but honestly, it doesn't matter what the size is. We've trained on 7 base pairs. It actually works almost better. It doesn't really matter. And it's also not that each motif is recognized by a single filter. It's kind of like the brain. It works, it learns very distributed information. So any kind of way the neural network learns it, it learns it. So that's why we don't go back and in the filter and identify where the motif was learned, but we're simply saying, okay, it had high prediction accuracy. Now, how important were the bases in each region? So we're going back to that. And this is where you see the motifs already appearing. And they're completely de novo appearing. So we're not based on any assumptions. So we're seeing those, but then we have to summarize those. And this is what I didn't explain in a lot of detail. So we're essentially collecting regions of high importance, and then that gets clustered. And it's a very memory intensive process because there's lots and lots of information, but it's simply just a clustering algorithm. And then you can use that to summarize motif representations in a more traditional way, like a PWM, or we can also summarize in what the network thinks is important. So this is nice, like a base that was really of high importance will get more weight in the motif representation. So this is kind of cool. So it's a different way of representing the motifs in a more network way. But other than that, it's really extracted secondarily to the actual predictions. So this is where the interpretation comes second to the predictive accuracy. I know it's a switch way. I call it the inverting paradigm.
Yes, you have to think like inversely. It goes
Exactly.
the other way. And then we are traditional geneticists and I'm developmental biologist. So I was thinking, I have another question that could be very naive too. Can you use, I don't know if you probably know that you can modify the enhancers and then, or change the enhancers and then you move the band from one place to another in Drosophila. So you get, I don't know, even a skip and then you mutate something and then you get a big band or a narrower band, or you move the band from one side to the other.
Yes.
So could you use these kind of neural networks to predict the actual phenotype of or the expression domain of your gene? Because that would be great. You can probably predict morphological changes or things like that.
So it's sort of a different task because you're sort of predicting a pattern. And so you can use neural networks for that. I know Saurabh Sinha, for example, did that. In our case, the readout is genomic information. Unfortunately, it's not. I wish we had single cell resolution. Right now, we're taking entire embryos. We don't have that resolution on where dorsal is active in the embryo. So that's kind of for the future. But we can predict whether a mutation will result in a defect. So even though we don't know what happens to the expression pattern, whether it's shifted or more narrow, but we can predict whether that base was important. And that's already an advantage. So if we're seeing, let's say, there is more than what we expected, for example, snail, we're seeing like, oh, there's a motif that previously people didn't consider as being important. We can mutate that and then see what happens. So it's still helpful, our model, but it's not at the level of predicting stripes yet, because we don't have the single cell resolution. Yeah.
Thank you very much. Very nice talk.
So there is another question in the chat. Actually, this comes from YouTube, from Julio Collado.
Question is, do you have a way to learn how is the motif built, or is it a black box as neural network models usually are? How the motif is built? Well, it's really just a filter. So it's that one hot encoded sequence. So it's basically you have for each base, you have a 1 or 0. That's really what it is. And then you're scanning and looking for things that produce a pattern that's predictive in the output. And that's how you learn motifs, in fact. And that's one of the earliest application of neural networks. It learns that really well. But as I mentioned before, the problem is that it's not necessary. So this is a filter is one of those sort of units that will scan. And you're using many sort of to increase your probability of picking things up. But you also make your model more complex the more filters you have. So you want to keep it minimal. And the problem, though, is that you really cannot predict which filter will find which motif. And sometimes you have multiple filters kind of detecting a motif, like a slightly different version of that, right? So the network is completely agnostic of what is 1 or 2 motifs. And so when you go in and actually just, go into filters, which people have done, yes, you do sometimes find those motifs, but it doesn't mean you've found the entire, you find all motifs. And so, Anshul at least says, don't even go in there and try to find that. So yeah, it can be done. But I think a better way is to systematically probe what the neural network, so use the whole neural network as, the trained network as a tool and really trace everything back or use everything to interpret in order to extract motifs. So that's sort of the answer. Yes, we know roughly how it's built, but I don't think that helps us to understand it. That's kind of the, so that a lot of people want to know, like, why don't you just open up the black box? But we don't, because there are millions of filters there. And each time you train a network, it might be learned in a different way. So it really doesn't help that much to open the black box. It's a better way is to go in and query the model with things that we expect might have learned and be really open-minded to the patterns that we see. So that's kind of where we are.
Okay, there is another question in YouTube, in the chat, from Victor Tarrafria.
I'm not familiar with eukaryotic species, but for the nanoc example, how do you discern two discovered motifs belong to, or just one of them? That's the really good question. In fact, we don't. And so in the case of nanoc, we did identify 3 motifs. And the reason we knew it was nanoc because they essentially, once you started to overlay and look at the chip nexus pattern, they actually started to resemble each other. There were sharp motifs based on the chip nexus data that were very similar. And so this really, by manually inspecting the actual experimental data, we were able to say this is indeed nanoc. We also validated there was a structure for nanoc that had a different motif and there was also one gel shift, and all of this made sense with the 3 motifs. But that's not always the case. Sometimes you will find different motifs, and you might not have that experimental knowledge to know whether it really is belonging to that same factor. So in general, I would say that is really a problem, or it could also be a motif of a related transcription factor. So if you don't have chip nexus data, you don't always have that information. I think that's one of the biggest drawbacks. If you just analyze ATAC-seq data, is a big problem because you often see this sort of aggregated motifs. I mean, even we saw motifs that were not very precise. Is it just one transcription factor? Is it multiple? You can cluster it, but is that accurate? I mean, you might see multiple, but it's really just one transcription factor. Or you might see one transcription factor and it's still bound by multiple. So yeah, this is a really good question. It's not trivial at all. But yeah, with chip nexus, you can. So.
Okay, I don't see any hands. So I'm going to, I have a couple of questions.
Yeah.
So enhancers are transcribed, right?
Yes.
When they are active.
Yes.
So and there's a particular, both strands are transcribed. And if I wonder if there is a particular position with the motifs in reference to the transcription start point of the enhancers, because I mean, if it's possible to find some indication that a distance is important, or maybe another kind of motifs, in order to allow the RNA polymerase to start transcription in relation with the motifs.
Yes, so I don't know. I've given talks on this, in fact. So we have data on that. We have a project on that in mammalians. I should say in Drosophila, we don't really see enough enhancer transcription. We've not been able to do this on a Drosophila embryo. I don't know why that is, but in mammals, it works beautifully. So when you use nascent transcript data or cage data, and when you analyze it, it's actually amazingly precise, the enhancer. So we see that the transcription start site has a very defined relationship with the nucleosomes that are flanking the enhancer, and that even the motifs are often preferentially spaced to that start site of the enhancer transcript. So the enhancer transcript does have also a motif, like it has a CA, that's the preferred start site in general, and we see that, but of course it's not always the case, and it can be very dispersed. But if you look for the preferred start site, you do see that CA. What else do we see? Yeah, we can also block with triptolide, for example, and we see that now we're seeing the initiation of that enhancer transcript, and it's moving kind of inside. So yeah, this is, hopefully we'll get this out soon. This is very interesting, and unfortunately I can't give you an entire talk on that.
That's okay.
But I think also other people have seen this now, that enhancers, they look much more like promoters essentially, except for that the transcripts are much less stable. But the overall patterns are not that different from promoters. Yeah.
Okay, thank you. The other question is, I'm curious if you have tried to analyze the OX genes, I mean, the binding of the HOX proteins in enhancers, because as you know, the HOX binding domain, the sequence that the HOX genes recognize is quite not the very, there's no consensus sequence at all.
Yeah, that's true.
HOX genes require other proteins to really fix in a particular position and then activate transcription. So then I'm curious to know if you have analyzed that.
Yeah, we have not. So we've collaborated with Rob Krumlov on his Hox binding data. In fact, so Boni Kumar, he used ChIP-nexus for one of the Hox transcription factors. The problem was that his coverage wasn't high enough, so we couldn't train VPNet on that.
Okay.
So we haven't gone back to that. I mean, we initially looked, but it wasn't promising enough for us to pursue it. But I agree, it would be a really good way of extract, because Hox gene binding is extremely complex, with the cofactor, the MEIS, and what's the other one called? So I think the VPNet would be a great way. As I said, the sort of strict motif spacing is really done well by VPNet. This approach would be really perfect. Yeah, but it needs more systematic data for that.
Okay.
Yeah,
okay.
But again, it's kind of not enough. If we had more experimental data and more people willing to train on that, we could do so much more.
Okay. Yeah,
this is definitely a problem that's well ripe for that.
Okay, just to finish. So VNet also predicts super enhancers. The motifs is super enhancer. Yes.
So we've trained on 1 KB regions, right? So we're sort of ignoring the bigger context. So if you wanted to learn, I assume you mean the super enhancers that are multiple kind of smaller enhancers that are cooperating to produce an output. And so we haven't looked at that. So you would have to look at more longer range models. And so there is obviously cooperativity there. When people have dissected it, it seems kind of complex, like sometimes it's additive, sometimes it's synergistic, but it's hard to predict exactly what that is. And so when people have analyzed high-C data, I see sort of the same problem, that it's not so easy to see those patterns of the synergy. Hopefully progress will be made in order to do that. We have not specifically taken that approach. So we've always kind of felt we wanted to go really high resolution. And so the longer you use regions, you'd have to kind of bin a little bit more. So you're losing the resolution in that approach. And so we have not done that.
Okay. Well, I think that I was the last to ask, the last person to formulate questions. Well, thank you very much, Julia. So wonderful talk. So we learned a lot.
Thank
you.
I
hope to see you soon in person.
Yes, that was wonderful. Thank you. Hopefully better. Hopefully it's a little bit better without.
No, actually you did very well.
Thank you.
We didn't notice anything.
Yeah. Okay. Well, thank you.
Okay.
Good day. Hopefully Mexico won today.
Okay.
Bye bye.
Bye bye. Gracias a todos. Ok. Fue el último seminario del año.